Chapter 12, Section 2 begins with pedigrees, which are just diagrams that show how a trait is inherited over several generations. So pedigrees can be used to track pretty much any genetic characteristic that you want to look at over through the years of a particular family, but you mainly are going to use them to track genetic disorders. So in this pedigree here, this is showing 3 generations, and they keep generations on a single line. So like this would be the grandparents, for instance, this could be the parents, and then this generation would be the grandkids. Now here they show a little more details than some of the ones that we will use. Ours will look more like this one on this side with circles and squares, and then you can see that some of them are filled in, some are not, and some are half. Circles are always representing females, and the squares will represent males. If they are an empty circle or square, then that means that they are unaffected by whatever trait you're tracking. If they have a filled in circle or square, then that means they are affected by that disease. And if they have a half filled in circle or square, then that means that they are a carrier. So the other thing that you're seeing on this one here, this slash, that means that it is a deceased person. So you can also represent that. And there are other symbols that could be used, but the main ones you need to know are the circle and the square, and then filling in versus an empty or a half-filled circle or square. These ones are representing like a couple, and then this couple's children are branched off from there, and their direct children are attached to that line. So they would have a male that's affected, a female that was not affected, a male that was affected, and another male that was not affected. And then their spouses would be attached to them separately. We're going to look at some patterns of inheritance. Some of these we've talked about already, but these are going to be the next 6 patterns that we look at. So how things get passed down from generation to generation. The first one is a single allele trait, and these are ones that are controlled by a single allele of a gene, so either a dominant allele or a recessive allele. Some examples are Huntington's disease. This is an autosomal dominant trait, so it's passed down through your autosomes, and as opposed to an X-linked or a Y-linked trait, and it's a dominant allele that causes this. So you only need one of these dominant alleles in order to have this disease. Huntington's is a degenerative brain disorder, and one of the issues with this is that a lot of times people don't start developing symptoms until like late 30s, early 40s, and by then a lot of times they have already had children, so they might have passed that trait on to their children without necessarily knowing it. There are ways to detect things like this though. You can get genetic screening done, and they can test for what's called a genetic marker, which is this short segment of DNA that's closely associated with that particular gene. And if you test positive for this genetic marker, that can tell you your chances. Usually it's a 96% chance that you have that gene for the disorder. Cystic fibrosis is another disorder that is caused by a single allele, and this one is caused by an autosomal recessive pattern. So in order to actually have this disease, you have to have both recessive alleles. And cystic fibrosis is a lung disorder. The second type of pattern of inheritance is a multiple allele trait, and this is when you have 3 or more alleles for the same gene that code for a particular trait. So for instance, our blood types, we can have A blood, B blood, O blood, or AB blood. And there are 3 different alleles that cause those particular blood types. You can have an A allele, which is represented by a capital I and a capital A exponent. A B allele, which is also dominant, or a lowercase I, which represents the recessive allele, and that would give you type O blood. So you still have 2 of these alleles that will combine to give you your genotype, but there's 3 possible alleles that could contribute to that genotype. So here are the possibilities. If you have type A blood and you're homozygous for that, you would have 2 big A's. If you're heterozygous, you're going to have a big A and then a recessive allele. For B blood, it's very similar. You also have two options, the IB, or the B and the recessive allele. For type O blood, there's only one possible genotype. You have to have both recessive alleles. And for type AB blood, you have to have a dominant A and a dominant B. That's the only combination that you could have and still have AB blood. So here's a type of problem that we might look at with blood types. It says a mother gives birth to type O child. The mother's blood type is A, and there are two potential fathers. One is type A, and the other is type AB. Which one is the father? So if the mother is A and the child is O, the mother must be the heterozygous version of A blood, meaning that she has a dominant and she has a recessive, because the child is the recessive blood type. So the child would have to get a recessive allele from the mom. Now, if we set up some Punnett squares here, here's our mom's genotype, IA, lowercase I, IA, lowercase I. One dad could be AB, and the other could be type A. So if the dad's AB, these are all the possible outcomes here, and none of those would have type O blood. So the father that's AB is really not the father. Now this one, if the father's type A blood and he's heterozygous, then there is a 25% chance that their offspring could have type O blood. So this guy could be a possibility. Now if he was homozygous for A blood, meaning that he has two dominant A alleles, then this man wouldn't be a possibility either. It would have to be someone with at least one recessive allele. Another form of pattern of inheritance is polygenic traits, and these are traits that are controlled by two or more genes. So not just more than two alleles, but two or more genes. And most human characteristics are going to be polygenic, which allows us to have a range of phenotypes. So some examples we've talked about in the past: skin color and eye color, height. We're not just one category or the other. We're not all 5 feet tall or 6 feet tall. There's a whole range of phenotypes that we could be. So generally when there is a range involved, then they're going to be polygenic traits. We also have to consider that the environment can play a factor too. Those are called complex characters, when both the genes play a role and also the environment. So for instance, you may have genes to, grow to be 6 feet tall, but if you don't eat the proper nutrients growing up, you probably won't ever reach that maximum capacity. You could also perhaps have a sickness that could cause you to not fulfill your full potential, or maybe an injury. Okay, so there are other environmental factors that can play a role as well as your genes. X-linked traits are also a form of pattern of inheritance. These are going to be traits that are controlled by genes that are on the X chromosome. So such as color blindness, which we mentioned before. This is an X-linked recessive trait. The most common form of color blindness is red-green color blindness. The muscular dystrophy, which is an X-linked recessive trait as well, and that is a degenerative muscle disease. And then hemophilia is also an X-linked recessive trait, and this one is a blood clotting disorder, which the diagram that I have up here, this pedigree, shows one of the ancient Russian families, and it was very prevalent then. There was a very small gene pool, and they didn't like to bring in a lot of new people to marry into the family and make that gene pool larger. So they started getting a lot of these recessive disorders affecting their males especially. You're going to see X-linked disorders appearing more often in males than females, because females have two X's, so they have an extra chance of getting a dominant allele that will help to mask the recessive, whereas males, you either have it or you don't have it. Sex-influenced traits you'll need to know are different than sex-linked. Sex-linked means they're actually genes that are attached to the X or Y chromosome. Sex-influenced traits are shown by the presence of sex hormones such as estrogen and testosterone. So those hormones will influence the expression of certain traits. So for instance, pattern baldness, it's actually caused by a dominant allele in men and a recessive in females, so that plays a role. But then also it's influenced by testosterone, which men have more of than women. So we see this happen more in males than females. Finally, there's non-disjunction, and non-disjunction is where the chromosomes don't separate out properly during meiosis. So one gamete ends up with too many chromosomes and the other with too few. That results in either a trisomy, which is when you have too many copies, 3 copies of a chromosome instead of 2, which will in humans result in having 47 chromosomes instead of 46. Or you could have a monosomy, which is when you only get 1 copy of a chromosome instead of 2. So that person would end up with 45 chromosomes instead of 46. And then these are all some disorders that could happen with non-disjunction. You could have Down syndrome, which we've talked about before, which is trisomy 21. So there are 3 copies of your number 21 chromosome. Klinefelter syndrome, which is when you have 2 X's and a Y chromosome. And then Turner syndrome is a monosomy. That's where you have an X, and that's it. There's no other X, there's no other Y. And both Klinefelter's and Turner syndrome have a range of phenotypes similar to Down syndrome. You can be highly functioning or low functioning, depending on where you fall on that spectrum. Some Klinefelters are males, and Turner's syndrome people would be females, but many of them end up having mild to even severe mental retardation. They can have infertility problems. And then others, you might not necessarily ever know that there's anything out of the ordinary with them. So it just depends on where they fall on the range of phenotypes. And finally, there's a couple different times that you can detect these disorders. You can do it before a pregnancy, during a pregnancy, or after the baby is born. Before pregnancy, people can get genetic screening done, and even some throughout pregnancy. And basically what you can do is just go in, get samples of blood and DNA, and you can have karyotypes done where they can then tell you chances of passing on certain disorders. And that's where the counseling portion comes in. That's really where you're told basically your odds of having disorders or not having those disorders. During pregnancy, they can do an amniocentesis or chorionic villi sampling. Amniocentesis is when they take a sample of the fluid from around the baby, and the chorionic villi sampling is when they take a sample of the tissue that grows between the uterus and the placenta. And with both of those samples, they would then form a karyotype and do much of the same testing that they could do with the genetic screening process. And then finally, after birth, they can do more screening on the child and see if there's any sort of issue that child might have. One disorder that they can screen for is called phenylketonuria, or PKU, which happens in about 1 in every 10,000 babies in the U.S. And that's where your liver can't break down the amino acid phenylalanine. And if untreated, it can cause mental retardation, it can cause central nervous system damage. But if it's caught early enough, they can prescribe you a special diet, which is very restrictive. But if you follow this diet, then you end up not having any issues with this disorder. So there are some benefits to being able to find out quickly whether you would have this disorder or not.